So what I'm going to do now is switch over to the document camera here and begin the lecture on the cell. Quite often in teaching this graduate level course, I'll have students that ask me to serve on their examination committees. At the master's level, the comprehensive exam committees. Too late now, but during your dissertation defense, maybe I'll have a chance to come after you. For the doctoral level students, the written exam is generated by a group of faculty, the Doctoral Studies Committee. I don't know if any of these questions would show up there, but certainly this is a level of expertise that you should have at any level of graduate studies, and to be able to just grab a piece of paper and a pen and describe the cell, subcellular organelles, and how they're put together and how they function in the cellular activities, certainly protein synthesis and how that relates to the biosynthesis and degradation of any other biological macromolecules, and be able to draw higher magnification and higher magnification levels right down to the molecular level even to explain this information, whether you're teaching or just working in a research lab trying to share ideas and information. So I go through this each year in this course just to show that I still need to practice those skills myself every now and then. So the typical approach is to draw a composite cell, and it will be a cell that has a nucleus, so a eukaryotic cell. And I'm not getting a large nucleus here because I'm used to doing this on a chalkboard. And of course what you're recognizing is that there's double lines here with little circles as we're going around, whereas surrounding the entire cell is just a single line. So this is the cytoplasmic membrane, as we know. And the cytoplasmic membrane, again, as we know, encases or contains the cell itself and separates extracellular from intracellular compartments, anatomical compartments, certainly fluid compartments. There's extracellular fluid and intracellular fluid that become very important. Obviously, the endocrine system, neuroendocrine system included, is involved in controlling the balance between extracellular and intracellular fluid compartments, the ionic content, and a variety of things like that. So we will end up focusing a great deal on intra and extracellular fluid compartments, as well for the methods of communications between the endocrine cells, the cells of origin, and the target cells, and how that signal is transduced across the cytoplasmic membrane. The nucleus has two of these membranes, so it's called the nuclear envelope, as we know. So the nuclear envelope is two of these membranes, and in a moment we'll zoom in on the cytoplasmic membrane to illustrate it, but we remember that it is a lipid bilayer. So this is a doublet of lipid bilayer membranes forming the nuclear envelope. So this means that there's an anatomical subcompartmentalization of this junction between the intranuclear and extranuclear or cytoplasmic spaces, and it indeed is a very important area. It's not just this void where nothing happens. We don't know a lot about what happens there, but we do know that there is a lot that does happen in this interleaflet compartment, if you allow me to call it that. And then we know that these are the nuclear pores. And what's the significance of the nuclear pores? Any type of opening or portal is going to allow movement of molecules. Molecules will move out of the nucleus, and molecules will move into the nucleus. Inside the nucleus, what are the major things that we find there? There's the chromosomes with the DNA. And we'll zoom in on the DNA in a while and kind of review it a bit. I can't draw the double helix too well, but we'll try to remind ourselves of that. What else is located in the nucleus associated with the DNA? General biological macromolecule would be proteins, right? So a variety of proteins are present here. Are proteins synthesized in the nucleus? No, we know that they're synthesized in the cytoplasm under the direction of the nucleus. I'm not suspecting that you don't have that in mind. But this brings us to the point that the proteins, I'll just put an asterisk on these, saying these are proteins that have permission to access the nucleus. So they're synthesized in the cytoplasm and must move through a pore, nuclear pore, to gain access to the nucleus. Now that may not be true for all of the proteins that are there. We know that during cellular mitosis, the nuclear envelope disappears, and a lot of the proteins, the histones, and a lot of other associated proteins interact with the DNA, bind the DNA, and twist it and all that stuff, and very likely stay there for a long, long period of time and may not be replaced. I'm not sure what the turnover of histone proteins are in a cell. We know that not every piece of DNA in this cell, if we think that think of it as a neuron of the neuroendocrine system, is used to ultimately to produce protein. Not all the genes are expressed. So some of the DNA is wrapped up very tightly and unavailable for gene transcription for the lifetime of that cell. So we have to ask ourselves, are those proteins there forever and they never have to be replaced? We don't know. I don't know anyway. I don't know that much detail about the histone proteins. But I do bring it up because a big part of what we discuss in this class is the life cycle of a biological macromolecule. It's essential to think of that when we talk about any type of biological signaling mechanism that's dependent upon a chemical. If the chemical stayed around forever, you wouldn't have a signaling system, because it's always there, always there. My hand stays here all the time. There's no changing information, so you don't realize anything's happening. It's only when it appears and disappears that we can realize that something is happening. So we all know that chemical molecules that are carrying a signal are produced. They're sent to a target. When the target receives them, it realizes that something has changed, responds to that, and then that signal goes away, and it knows that the change is not essential anymore. That's the key molecular aspect of homeostasis as we understand it. So we'll zoom in on DNA and its proteins and that in a while and focus on it a bit more. What we do know is that there's something else in the nucleus that has at least the N and the A, but not the D, and of course that's RNA. And RNA, especially the messenger RNA that we'll focus on, is a key molecule that exits the nucleus. Of course, ribosomes are assembled somewhere and are made up of ribosomal RNA and the ribosomal proteins. So we know that ribosomal RNA is exiting the nucleus as well. And what structure is associated with the production of ribosomal RNA? That would be the nucleolus. So usually there's a big dense thing in here called the nucleolus. I'm not going to label it, but you know what it is. Then scattered throughout when we're looking at a nucleus, we see areas of dark substance. There's one other thing that we didn't mention in here, and that, of course, is the nucleoplasm. Let's not forget the fluid component of this. It's not all dry proteins and dehydrated DNA. It's all hydrated, and it's in an aqueous solution, just as is the cytoplasm. So there's the nucleoplasm. Got to carry in the nucleic acids to repair DNA if we're going to be doing that. Certainly the ribonucleic acids have to be carried in for the assembly of RNAs. So there has to be a fluid phase, an aqueous phase, to get things in and out. So other than the nucleoplasm, there is the chromatin, as we know, that represents the DNA associated with the proteins, in essence. So there's two types of chromatin. There's euchromatin and heterochromatin. And the heterochromatin is the darker, and the euchromatin is the lighter color. Whether you're looking at a nice high-resolution light micrograph or definitely an electron micrograph. So if heterochromatin is darker, do you think it's associated and compacted tightly with proteins? Why do I always ask a question that has the answer in it? Right? So heterochromatin is basically inactive DNA, right? It's not actively transcribed. So in a sense, that's the silent DNA in a cell. And then euchromatin is lighter, meaning that it's uncoiled and it's stretched out, and the DNA is available for access by the appropriate proteins to transcribe the DNA into the RNA. So the concept that I'm building on here, as we're introducing the idea of the DNA, gene, and gene expression, is differential gene expression. And this differential gene expression will be the focus of the lecture, the next lecture. What I want to do now is just quickly, in the next 4 or 5 minutes, is to fill in the rest of the subcellular organelles. We obviously have talked about the nucleus that contains the DNA, the encoded information that is transcribed into the ribonucleic acid, slightly different version of that encoded message. As we know, there's a lot that happens between reading the DNA and having the messenger RNA ready to be shipped out to the cytoplasm. I used to call it post-transcriptional processing, and have been corrected by cell and molecular biologists to refer to that as RNA processing. We will discuss that as well. When the messenger RNA enters the cytoplasm, it has one molecular target, which is what? What subcellular organelle does it target? Not like it goes hunting it down, but anyway, the one that it can interact with, of course, is the ribosome. So we find ribosomes structurally in subcompartmental, two different types of subcompartmental areas within cells. They can be free ribosomes, or, as we know, they can be intricately associated with a reticular network of tubules and saccules called the endoplasmic reticulum. The ER, when the ER is associated with ribosomes, and these used to be called fixed ribosomes, but they are not fixed ribosomes. They're ribosomes that are clustered in a close proximity to portions of endoplasmic reticulum. When we find these ER with the closely associated ribosomes, it of course is called rough ER, or for short, the RER. So we know that there's one type of protein produced at the free ribosomes called the housekeeping proteins, like the enzymes for the glycolytic pathway that are in the cytoplasm. Some of the proteins that come from nuclear DNA genes and end up in mitochondria. So there's a wide variety of housekeeping proteins other than the ones that end up going into the ER as they are synthesized. So as the messenger RNA would interact with a ribosome close to an ER strand, it'll interact and bind to the interact with and bind to the ER membrane, and then the protein, as we know, ends up being pushed into the endoplasmic reticulum as it is produced, or this is the process of translation, as we know. We'll see all this again, and I know hopefully it's a very familiar review for you. So these proteins get shuttled to a point where these endoplasmic reticulum membrane buds off, goes to the Golgi apparatus, as we know, and quite often these will end up being secreted by the cell, or the proteins will be inserted into the membranes, and as we get this secretion process, exocytosis, the membrane proteins end up in the membrane of the cell. So these little proteins would be in the membrane here as it's going along, all through here. Other of these vesicles will become lysosomes, so there's degradative and corrosive enzymes that we know about, peroxidases and things like that. I always include peroxidases, but it's actually more complex. Peroxidases are produced by free ribosomes or in the cytoplasm, become incorporated into lysosomes when they're needed. So we are generalizing with this generic cell, and I'll stop there, and that's a pretty good introduction to how protein is produced within a cell and what are the different basic categories of proteins and where are they produced and where do they end up. So we'll begin the next lecture focusing on the cell membrane, different types of proteins in it, and we'll then shift over to differential gene expression. Thank you for your time and attention.